It is often thought that tissues within our organs are a tight compaction of cells, where in actual fact they are usually more spread out. Between the cells is a complex support network comprised of proteins and carbohydrates called the extracellular matrix, the ECM. The main role is to protect and support cells, like the way items are protected during delivery by packing them with wads of paper or polystyrene scraps. Yet this is where the analogy falls apart. The ECM is a highly complex and provides many more functions than just protection. These include providing a scaffold to regulate correct function and shape of the tissue, influencing the cell's behavior in accordance to the changing environment, and regulating cell-to-cell communication between adjacent cells. Just as the tissues within our body differ, so do the components of the extracellular matrix. Each composition is adapted to the needs of the environment. This can be hard and calcified to form bone, or soft and elastic as we find in our lungs. But how does the ECM know which components to use in which tissues at which time? The components are actually produced by local cells. They secrete specific products, defining the specific characteristics of the ECM to best fulfill their role in the tissue. In most tissues, ECM ingredients are secreted by cells called fibroblasts. However, in more specialized tissues, these fibroblasts come in different flavors, for example osteoblasts in the bone. Yet all ECMs share three common components: an extensive proteoglycan network in green, chains of cross-linked fibrous proteins in blue, and cell receptors called integrins in orange. Firstly, the proteoglycan network comprises of sugars called glycosaminoglycans. These long polysaccharide chains are attached to hundreds of core proteins, with each protein having many similar smaller carbohydrate groups attached. They exist as a compressive filler substance to protect cells, as well as storing many cellular growth and immune activating factors. Secondly, the fibrous proteins are embedded within the proteoglycan network to form the basis of the structural scaffold. They also help attach the ECM to other nearby cells. The two most common constituencies of this group is collagen and fibronectin. Collagen is a strong triple helix protein comprised of two identical chains and another that varies only slightly. The variation of one of these chains defines the type of collagen this is and gives it slightly different properties. The collagen fibers are interwoven within the ECM to form a basket weave. Fibronectins come together to form insoluble fibers between cells. They bind collagen and integrin on the surface of local cells to allow stretch-mediated cell-to-cell communication. Finally, integrins are cell receptors that span the membrane. Within the cell, these receptors bind to microfilaments to convey stretch and pull messages to the cell to change behavior and respond to the local environment. On the outside, they are connected to the ECM to also allow cells to send and receive messages. These integrins are also classed as part of a group known as the cell adhesion molecules, and help cells stick together in the ECM and hence together. During contractions or movements of the extracellular matrix, the integrins rearrange the internal microfilaments, which send signals to the nucleus to turn on or off certain genes which allow the cells to adapt to the behavior of the changing environment.